Okay, hello everybody. This is going to be the last video for this week of content. For 4 videos today, so through a lot of work. But yeah, we're going to be talking about oxidative phosphorylation, so basically just ATP synthase and some other aspects of that time. So let's go ahead and get started. So I'm not going to go over this, but the main idea is that, we get some electrons from citric acid cycle, and then these go through oxidative phosphorylation. We make ATP, and then we do all sorts of stuff, right? Mitochondria, they use proton gradient to turn into ATP. Cool. Not much else there. That's important. We already talked about this proton gradient, right? So where NADH, right, goes through complex 1, 3, and 4, while complex 2, well, which is not shown. Complex 2 would be somewhere in the middle here, right? And FADH2 goes through that and only produces 6 protons versus 10 for NADH. The proton motive force. So you don't have to know any of this confusing math, but I think the way that you should keep this in mind is just it's the energy stored in the proton gradient, right? So basically, the more H pluses you have on the more H pluses you have on this side, right, the easier it's going to be for these protons to go down the gradient for ATP synthase. So that's the proton motive force, right? Basically, how much do those protons want to travel to the other side? And if you have a lot of H plus on this side and none on this side, you can imagine that it's going to be a very large driving force. You're going to have a very high proton motive force. So generally speaking, the proton gradient and ATP synthase are tightly coupled. And electrons just don't flow if the pH gradient is too steep. So what does that mean? That means that if there is very little H plus on this side and a ton of H plus here, then the proton pump is just not going to function. And that should make sense intuitively, because if there's less H plus on this side, right, the chance of these proton pumps even finding H plus in the first place is very low, right? And so they have to go down ATP synthase to replenish the H plus concentration on this side before they go back up, right? That's the main thing. So the idea here is that, as well as that, electrons are just not going to flow if that pH gradient is just not there, because they're going to try and drop the energy off here to pull a proton up, but there's going to be no protons, so it's going to get stuck, right? And we'll talk about this F0, F1 in a second. These are the different subunits of ATP synthase. They generally do not test on the experimental approaches for studying oxidative phosphorylation, but they do test you on the different inhibitors. So, example is DNP, dinitrophenol. We're not inhibitors, but it's an uncoupler. It's a decoupler, right? So basically what this guy does is, the proton gradient, right? The only way for the proton gradient to be reestablished, right? So after the proton gets to this side, the only way for it to go back to the other side is through the ATPase, which is going to generate ATP if it goes through there, right? But with things like DNP, what you can do is, once you generate the gradient, or once, when that gradient is there, the DNP is actually just a carrier that takes the H pluses from a high concentration to a low concentration. So basically, it does the same thing as ATP synthase, but it doesn't produce any ATP. It allows H pluses to go from high to low, but does not harness that energy, and instead just causes a loss of energy and a loss of ATP. Right. There's other uncouplers as well. You got FCCP and CCCP as well. But generally, the idea here is that they disrupt and they dissipate the proton gradient by acting as transporters to allow H plus to go from high to low concentration without having to go through ATP synthase. Just a cool little slide about why it was bad. People actually ended up using it to lose weight, and they actually ended up dying and having a lot of heart problems. So just a cool little slide. There's other types of inhibitors. We got oligomycin and venturicidin. I think I butchered that. Venturicidin. they both inhibit ATP synthase. This is ATP synthase. This is different from the uncouplers. So the uncouplers, right? They instead of directly inhibiting ATP synthase, all they do is they just help dissipate the proton gradient. But with these guys, they actually just inhibit ATP synthase. I want to make this clear that, you know, inhibiting ATP synthase has no effect on electron transport chain, right? Because ATP synthase is a separate protein that's not really connected to the electron transport chain. They kind of work together, but they're very separate from each other. So to inhibit ATP synthase, you can imagine that even if you use a bunch of electrons, put a bunch of H plus into the intermembrane space, none of it's going to go through and make ATP because you just blocked this channel off, right? And here's just a nice demonstration of chemical uncoupling. So you can see here, like looking at O2 consumption, right, and ATP synthesis, right, which is this reddish line here. So you can see that when we first start, right, we've got, you know, we just add pyruvate, right, and there's no ADP PI, but when we add the ADP and PI, right, you can see that the consumption of O2 and the ATP synthesis goes up steadily. But then when you add the ATP synthase inhibitor, right, it just flatlines. ATP synthesis flatlines, and then O2 consumption also flatlines. However, when you add the decoupler, right, ATP synthase is still flatlining, but the oxygen consumption is way up. So that's how you can tell whether it's an uncoupler or if it's just an inhibitor, right? Because if it's just an inhibitor, right, you're not really going to get like a... It's going to be a flat ATP synthesis, so there's not going to be any ATP synthesis, right, because it's inhibited, right? And there's not going to be really a lot of O2 consumption, right? And that's different if you use a decoupler, because when there's a decoupler, right, that doesn't affect ATP synthase in any way, but it does mess with the proton gradient. And so now the electron transport chain has to work 10 times harder to actually generate that proton gradient. So you end up getting this massive increase in O2 consumption and just a stagnant ATP synthesis where it just does not change. And here they just give you more examples of what this looks like, right? So if you have a buffer alone, just the buffer with mitochondria, we're looking at oxygen consumption again. If you add pyruvate, it goes up. If you add ADP, right, it goes up drastically, right? And then if you add the inhibitor, right, it drops back down. If you add the uncoupler, it goes way up again, right? And then if you add antimyosin again to block the chain, right, it just goes down to 0 pretty much. I've already mentioned this before, so I won't really dwell on it for too long, but every NADH 2.5 ATP and every FADH2 is 1.5 ATP. It's because it's 4 H plus per ATP, right? And NADH releases 10 protons, right? While FADH2 only releases 6, right, into the intermembrane space. That is why. And, you know, if you uncouple it, of course they can change, right? Because now there's a completely different amount of protons required since the uncoupling has messed with the ratios of protons going up to protons coming down, and they're not going through ATP synthase as much as they should. So mitochondrial uncoupling. So there are some like cells, right, that, you know, are generally just uncoupled to begin with. An example of this is brown fat. So brown fat cells, or just fat cells in general, right, especially brown fat cells, actually don't use, I mean, they use ATP synthase, but it's not quite as coupled as another type of cell. Instead, they actually use that proton gradient to generate heat, right? And that heat, you know, keeps the animal warm, right? So they have a ton of mitochondria, right? And that's what makes it brown, right? Mitochondria are generally brown, and it, you know, it makes it brown, but if you have a ton of mitochondria. So here we have our uncoupling protein. So these brown fat cells have this uncoupling protein, UCP1, and it does the same thing as ATP synthase. This is pretty inaccurate, actually. It doesn't just eliminate it, right? There's still some H plus going through this to make ATP. It's just not as much as if this protein was not there, right? But basically, right, ATP synthase harnesses the energy produced by the gradient to make ATP. This one, the H plus just goes down and releases as heat, which warms up the animal, or warms up the baby, right? And here you can look at brown fat versus liver mitochondria, so tightly coupled versus uncoupled. You can see if we have the buffer, you have mitochondria, right? Oxygen consumption is pretty low for both. If you add pyruvate, you can see that the oxygen consumption here is massive compared to this, way bigger, right? When you add the substrate for ATP synthase, you can see it still produces ATP, but not really as much, right? And then if you look at oligomyosin, right, you inhibit it, right? You're still producing a bunch of oxygen, like still having a lot of oxygen consumption, right? Because UCP1 is still functioning, right? UCP1 is still dissipating the proton gradient to generate heat, right? And then if you add the uncoupler, right, then we see this big spike for the oxygen consumption rate in the tightly coupled one, but it does cause an increase in the one that's not that coupled, but it's not as much of an increase from the baseline E as it's compared to this E over here, right? And then anti-myosin obviously drops it all the way back down to G, just breaks electron transport chain itself. So this should hopefully make sense, right? What I'm trying to say is, when you add an uncoupler, right, or if you have an uncoupler in your cells, the oxygen consumption rate at a basal level is already much higher. The base consumption of oxygen is much higher than if it's just a tightly coupled, no extra uncoupling protein available, right? So the question is now, how do we actually capture that energy to make ATP? We know that it's turned into ATP, but how is that happening? And it's happening through this rotary freaking machine, right? It looks like a wind turbine almost, or like a plane engine. And this is actually really cool. I actually really like these images, like the images of crystallographies of the ATP synthases. So, all right. So if we go ahead and take a look at the ATP synthase, we've got a couple of different complexes that we got to look at here. We've got the F0, and then we've got the F1. And so the F0 is made up of the C ring, the A subunit, and this stator, which we don't really, the stator is not as important as the C ring into the A subunit. The F1 is made of the gamma, I think that's sigma, if I'm not mistaken, and epsilon, right? And so you've got these three alpha beta dimers as well. So these epsilon, sigma, gamma are like in this section here, but these alpha beta dimers are up here, and that's the important part. So we'll cover that in a second. And the other thing to keep in mind is the location of where these are. You'll see when we do Kelvin cycle and electron transport chain in plants, I believe it actually flips. There's some other stuff that goes on with the chloroplast that makes it a bit strange. So yeah, just something to remember because it's gonna show up soon. So you can see that the F1 subunit, or the F1 complex, right, is actually sticking into the mitochondrial matrix, right? While the intermembrane space does not have any of that, any of the machinery in there. So if we look at this, the cool thing about this whole model is that ATP synthase spins, right? But the alpha beta subunits actually don't rotate. What does rotate is the gamma, epsilon, and sigma subunits. These are what spin. I believe the C ring also spins as well. So that's something that we have to keep in mind when we're going through the questions here. So I don't really think that this is too important. It's there if you want, but the main idea is that you need to know that these guys spin, and these alpha betas do not spin. And another cool thing actually is that this actually does work in reverse. So the ATP actually can be turned into ADP and PI, which is really interesting. So we already talked about this, right? I think they were like showing a video here, which is kind of cool, but don't really care that much for me. But yeah, now here's where we get to the interesting part. So this is how the thing actually works. So we got protonation and reionization of glutamate in C-ring subunits drives rotation. So basically, the rotation of this whole C unit, the C unit here, and the gamma and epsilon here, is caused by the series of glutamate amino acids that basically act as proton acceptors and then donators. So I'm trying to see if they show a good image of it, but if we look here, this is supposed to be the membrane, I think. Essentially what's happening, right, is we have this glutamate and aspartate in the C ring, right? And so what happens is every now and then, right, a proton will get attached to one of the glutamates in the C ring. And when that happens, right, it basically leads to a spinning motion. And that spinning motion causes this to get to this position here. I'm trying to see how they did this exactly, because they're doing it in a bit of a different way than I'm used to, but essentially, right, you can see here, right, we start with an H plus in this section, right? Each of these like little H's here is a glutamate, right? And it basically goes around until it gets to this H plus section here, and it gets spit out the other side, right? And that's used to spin it. So you can imagine, right, that we have a glutamate in each one of these spots, right? And it goes all the way around. And so what happens is, for the first glutamate, right, it has some pKr value, right? And this H plus attaches to that glutamate. And then that glutamate donates its H plus to this glutamate over here, and then this glutamate over here, and then this glutamate over here, and then it keeps going. And each time it transfers the proton, a little bit of energy is released to power the rotation of this C ring, right? And eventually it gets to this H plus position here, where there's a glutamate here, and it then gets released into the matrix, right? And that's basically how we're using that H plus, the energy in the H plus, to power ATP synthase and generate ATP. But that's just how ATP or H plus goes through it. Now I have to talk about how it's how it actually generates the ATP. That's down here. This is just a cool little explanation for what happens, right? I wouldn't dwell on this too much. Yeah. This is what I would focus on. You definitely need to keep in mind the different subunits. So the alpha and beta like pairs. Alpha, beta, alpha, beta, alpha, beta. Right? So what's going on here is that the rotation of the gamma subunit. So this gamma subunit is rotated due to rotation in the C ring. Right? And so the C ring, with all of these proton movements, right? I should be specific here with it's not really a rotation. It's more of like the H pluses are rotating around, right? So these H pluses rotate around, right? They get transferred from glutamate to glutamate, and that causes the drive, right? But if you look here, the alpha beta kind of complexes, right? These they're not really subunits, but I guess they're subunits, but these little complexes, right? The gamma subunit, when it starts turning, starts pointing to different parts of the alpha beta like complex, right? So it points to one alpha beta complex and another, and then a different one, and it rotates by 120 degrees each, pretty much. So if we look here, right, we're gonna start with let's start with we start with this open guy, right? And basically, a rotation of this gamma subunit changes the conformation of these alpha beta subunits so that they attach an ADP, PI, and magnesium, right? And then another rotation happens, right? Which leads to another conformational change, which changes this ATP magnesium into this ADP, PI, magnesium into an ATP and a magnesium, right? And then that rotates again, right? And then this guy leaves, right? And this happens in all three subunits, or in all three alpha beta pairs, right? So you can see here, right? When this guy has this ATP, right, it rotates 120 degrees, and then it's now released. See? It's in a loose state. If it rotates again, right, that grabs an ADP and PI. It does that for all of these subunits, right? Essentially what's happening is that every time an ATP is released, a new ADP and PI is being attached to a different slot, right? That's the main thing I want to get across from this, and that's the main thing you should actually take away, honestly, maybe from this lecture is basically this is one of the big things that they could test you on for your exam, right? And so what you should know from this is that one full rotation generates 3 ATPs. Why? Because there are 3 different alpha beta pairs, right? And each one of them will generate an ATP upon a full rotation of this gamma subunit, right? So if we're trying to think about, how much ATP is made, we just think about how many protons are pumped per NADH, which is 10 protons, and how many protons per ATP synthase cycle. So there's 8 protons, right, generates 3 ATP. That's about 2.7 protons per ATP. So for 10 protons, we would expect a little bit more ATP, so something like that. But when we actually did the experiments, right, we found that every NADH yields 2.5, and then every FADH2 yields 1.5. So yeah. But yeah, and then here's the reason why some of that energy is not actually used to make ATP, right? There's a lot of co-transport systems that move ADP and PI into the mitochondrial matrix so that we can actually do this process in the first place, right? So if we take a look at this, right? I don't know why it says CO2. I feel like it should say O2. A bit misleading. But basically, right, if you go through the entirety of if you go through the entirety of this, right, if you just go from glucose to lactate, right, you're only going to get 2 ATP. But if you go all the way, right, through the electron transport chain, right, generally speaking, right, you'll get 32 ATP. And that's the main thing you should take away from this lecture. One of the main things, I said that like 3 times now, but that's the main idea I want to get across here is not only the functionality of the ATP synthase, but also just how much of a big difference it makes using ATP synthase versus just glycolysis. You're producing like 16 times more ATP by doing electron transport chain and aerobic respiration compared to anaerobic respiration. So it's a really big deal. But that's actually the end of this lecture. I feel like I sped through that one quite quickly, but hopefully it made sense. If you have any questions, feel free to stop by the IC or leave a comment or whatever. And yeah, hopefully the video is helpful, and happy studying.